And basically we would only be able to support half of the world's population if we didn't have synthetic fertilizers.
Wow.
So although it may look like I've moved away from medicine and wanting to help save lives, to some degree these simple things like fertilizer are actually more life-saving.
Hello everyone, and welcome to Mug of Science, part of Pint of Science Australia 2021. I'm Ravilekha Mossam, and joining me is Dr. Trevor Rapson, research scientist at CSIRO. Hi Trevor, how's it going?
Very well, thanks.
Good. So, our first question, what have you got in your mug?
So I'm drinking some strong black coffee, which
Oh, black coffee.
gets me going in the mornings.
Yeah? So you don't take milk, or is milk hard to get?
At home I drink milk, at work I don't drink milk because it's too much effort to keep the milk at work and keep it fresh.
Yeah, the sacrifices you have to make for science.
Yeah.
So, do you want to tell us a bit about what you do?
So I'm a biological chemist, so I work particularly on proteins that have metal centers in them, and the protein that I work on currently is a protein called nitrogenase, which converts nitrogen in the air into ammonia. And I'm very fascinated by it because this is a very, very difficult reaction. You may know from your high school biology that nitrogen is a very stable compound. That's why it's the majority component in our atmosphere. So basically nitrogen has a triple bond, and breaking that triple bond is very difficult. And so nitrogenase is something that nature's come up with as a way to do that and convert the nitrogen in the air into nitrogen that goes into the soil and into our plants.
Okay, so that's important for fertilizers, is that right?
So yes, we need nitrogen. It's quite a major component in our bodies and in all biological things. It makes up amino acids.
Okay, and
so in biology, bacteria, what we call, fix the nitrogen and convert it into ammonia.
Mm-hmm.
But then what we do with fertilizer is, if we want to get lots of plant growth, we have to add extra nitrogen in. So then you have to use an industrial process known as the Haber-Bosch process, which uses very high temperatures and very high pressures to break that triple bond and produce nitrogen. So the fertilizer is produced through this process known as the Haber-Bosch process, and that's then applied as fertilizer. But there's been a lot of interest in making ammonia for fertilizer, but with the green energy revolution that's taking place, ammonia has actually become very interesting for energy applications as well.
Oh, really?
So we're going back to trying to come up with new ways of making ammonia. And one of the problems with the Haber-Bosch process is it uses very high temperatures and very high pressures, so it's very energy demanding. And depending on the hydrogen supply that you're using, if you're producing your hydrogen from fossil fuels, then you're actually emitting a lot of carbon dioxide in the process of making the ammonia. So it's roughly, for every molecule of ammonia that you make, you actually emit about 2 molecules of carbon dioxide. So
Okay.
There's a lot of work to try and make the process better. And one of the sort of longer term goals is to actually go back to nature and to use the natural processes rather than using the industrial chemistry processes that were developed in the early 1900s.
Okay, wow. So we're trying to find biological ways of replicating this Haber process, which could be much greener, less CO2 and less energy needed.
Well, so nature already does it at the moment. There's soil bacteria that are sometimes associated with plants, like legumes, that are able to carry out this process. But what we're trying to do is trying to work out, can we use these natural processes in an industrial scale?
Okay.
So have them growing in a big bioreactor that we can then use to make ammonia. So rather than just relying on the bacteria in the soil, really try and ramp up the process. So we're looking at ways that we can take the biological processes and make them work at an industrial scale, so we can produce nitrogen and ammonia
Okay.
So we can produce ammonia from nitrogen at an industrial scale equivalent to what the Haber-Bosch process currently does.
So what do we have to do that? So you said these bacteria exist. So you're saying we can't just grow a giant vat of them and let them go. What do we need to kind of do to get this industrial process going?
So there's a whole lot of stuff that you need to look at. It's how to culture the bacteria, how to get rid of the products. One of the other problems is while the Haber-Bosch process consumes a whole lot of energy in terms of pressure, the biological process consumes a lot of biological energy in terms of ATP. So for each molecule of nitrogen that's broken, you need 16 molecules of ATP. So one of the things we're doing is trying to work out ways that we can reduce that ATP demand so that it's not such an energy consuming process for the bacteria. And ultimately what we want to do is have it so that you can have a solar panel provides the electricity, the electrons that drives the nitrogenase. So what we're trying to work on is how do we interface that, a solar panel, with the enzyme, the nitrogenase enzyme.
Oh, that's really interesting on the engineering technology and bacteria to work together.
Yeah.
So what does that creative process look like? Do you read, go for walks? Are you calculating things?
A mixture of everything. I think the biggest thing is just reading what's been done and what other people have tried. Talking to other scientists, and that's obviously been a little bit impeded by our lack of ability to travel and go to conferences internationally, but that conferences are really important for sort of getting people together and talking about ideas and brainstorming problems. Then I do I think often the best way to get creative is to get out of the lab and get out of the office. So I do try and build that sort of thing in. We're very lucky here. We've got beautiful mountain bike tracks on Black Mountain. So I'll quite often just go for a ride or something and often come up with good ideas while I'm out in nature.
Oh, that's nice. So how did you get into this line of work? What was your path? Did you always know you wanted to work in this particular area? What did you want to do when you were younger?
That's an interesting. So at high school, I wanted to be a medical doctor, and I wanted to do medicine. So then I went to university and did basically a pre-med degree. And the pre-med degree involved, it was originally going to be biochemistry and microbiology, but then I sort of got caught up in this chemistry side of things. And there was a particular professor who was really sort of inspiring and I found very interesting, and he was working in drug discovery for new chemotherapy drugs, and he sort of said, I caught this concept that if you're working at the interfaces of disciplines, that's where the most exciting things are happening.
Yeah.
So he was working at the interface of chemistry and medicine, sort of doing drug discovery. So I then sort of steered away from going into medicine, and I started doing chemistry research. So started off doing an honours degree where there's a large research component, and then went on to a master's. But I never really wanted to end up in the drug discovery space. I was always very interested in the protein side of things. So then I had the opportunity to come to Australia. This was all in South Africa. Come to Australia and do a PhD on a metalloprotein. So that's a protein with metals in, which is where I was really quite fascinated in. And what I'm really interested in is how the protein interacts with the metals and how apart they can't actually do things by themselves, but the sort of synergistic interaction is where this very interesting chemistry takes place. And so that was my PhD, and it still had a slight medical focus. And then I joined CSIRO in the health and biosecurity division or section. We now call them business units, but it keeps changing what we call them. And then I was focused on, again, a medical application. So that was with we're trying to make an asthma diagnostic. So it was a simple to use device that you blow into that measures your nitric oxide levels on your breath, and that can be used to predict the onset of an allergenic asthma attack. So it basically tells you your lungs aren't happy, there's some allergen around that's making them release nitric oxide.
Oh yeah.
So I did that for a while, but then I sort of increasingly got drawn into the energy space, and I've had a few projects that were sort of dabbling on the edges of energy, and then when this opportunity to come and work on the nitrogenase project came up, I jumped at it. And nitrogenase is basically the queen of the metalloproteins. It's by far the most complex. There's been years of study on it, and we understand little bits, but there's huge bodies of that we don't even understand. We don't actually even know where nitrogen binds to the active site.
Really?
So it's a very, very exciting area to work in, and that the reaction that it's doing is just so difficult. So I've really enjoyed moving into this area of nitrogenase, and it sort of links in with all the skills that I've already built. And so I moved away from the medical focus more into the energy focus. But actually, one of the interesting things is the Haber-Bosch process, or the ability to produce synthetic fertilizers, is actually the most life-saving invention that's ever been discovered.
Really?
And basically we would only be able to support half of the world's population if we didn't have synthetic fertilizers.
Wow.
So although it may look like I've moved away from medicine and wanting to help save lives, to some degree these simple things like fertilizer are actually more life-saving than vaccines and medicine. Not to downplay them, but we simply wouldn't be able to feed everybody if we didn't have these artificial ways to make fertilizer. So I am learning too that sometimes you can be involved in what looks quite different to science, but you're actually still having the impact in terms of people's life and health.
Yeah, there are many different ways to make an impact and help people.
Yeah.
So you mentioned that it's interdisciplinary, that you need all these different skills. We might think, material scientists, a geneticist, a chemist, they're all just scientists. But when you drill down to it, you're saying they have very different tools and different ways of thinking.
Not only that, different languages that they speak.
Yeah.
So like as a biologist, when you talk about enzymes, you talk about substrates. What's the enzyme substrate? Well, it's nitrogen.
Is the
That's what's
substrate
binding.
the thing it binds to?
The substrate is what binds to the enzyme and then gets converted into its product. So you have a substrate going to a product. If you talk to a material science about the substrate is the bit of glass or something that they're going to start building their device or their electrodes on. So you're actually using the same word for two completely different things. So it's in some ways it's quite challenging because you have to sort of learn to speak a common language that you can sort of all get on with and communicate. So you definitely have those sort of moments, and then sometimes your hardcore material scientists will want to use some chemical that's just insanely harsh on a biological system, and and then we work with people in Clayton who work on developing other methods of making ammonia, and all the units that they use are completely different to our units and completely different scales. So it takes sort of a lot of work, but it's still quite fun trying to help people understand, well, what are you bringing, what are they bringing, how can we actually sort of intermesh them. And I think particularly going from a biological system to a more industrial application, that is just two completely separate schools of thought about how it all works.
Yeah.
So it takes a lot of time to get everybody understanding what's going on and what are the potentials of both and what are the problems of both.
Yeah. I guess it's a skill to be able to talk to people in all these different mindsets and get them all on the same page. So did you mention you also do work in renewables and renewable energy?
So where it fits in, and I've got this diagram that I can show you. So you probably heard quite a lot about hydrogen energy as a big opportunity for Australia to export its renewable energy.
Yeah.
So essentially we could replace a lot of our combustion engines with hydrogen fuel cells, and the reaction between hydrogen and oxygen produces water
Instead of CO2.
instead of carbon dioxide. So it's a very, very green process. But then when you want to transport hydrogen, so you probably know that hydrogen's a gas normally, and to liquefy it, you have to put in a huge amount of energy because you have to really cool it to, I forget the exact numbers, to insanely cold temperatures to get it to actually liquefy. So if we're actually going to transport hydrogen from Australia to Japan, for example, you need some way of doing that, and the best way is as a liquid. But you can't use hydrogen as a liquid. But what CSIRO has developed is a special membrane and a technology that you can convert ammonia back into its nitrogen and hydrogen constituents. So, and this is a technology that Japan's very interested in. So essentially Japan wants to buy hydrogen from Australia, but we will ship it to them as ammonia.
Oh, is ammonia solid or a liquid?
As a liquid.
Okay.
And then the advantage of it is we already actually ship a lot of ammonia around for fertilizers, so the infrastructure is all there as a way to ship it.
That's interesting.
And the question is, can we just build that capacity? And so there's some demonstration plants, like there's been one that's been built in the Pilbara, and essentially what they're trying to do is, instead of making the hydrogen from fossil fuels, they're using a new process called water electrolysis, where you provide a current into water and you split it into oxygen and hydrogen. And so that provides the hydrogen. Then you get the nitrogen from the air, and then you put it through the Haber-Bosch process. But the idea is to have all of this powered by solar energy, and so that's a way of making green ammonia that hasn't had any fossil fuels, and that can then be shipped to Asia. And this is a pathway that's basically very mature now, and they already are starting to build plants so that we can actually transport green ammonia to Asia.
Oh, wow.
But then where my research comes in is we're going to try and make it even simpler. So instead of having to make hydrogen, nitrogenators don't need hydrogen, they only need nitrogen. And so we'd have a bioreactor that operates at ambient conditions, which would then be a much simpler way of producing the ammonia for the export. So my interest is in renewable energies, as making an alternative to the Haber-Bosch process to produce ammonia.
And then using that ammonia not just as fertilizer, but as fuel.
And then the other thing, so it's interesting you mentioned that this could also then be used, because it's a much simpler thing, you could look at having on-farm production of ammonia.
Really?
So we could have this sort of little way of, so farms can have their own little fertilizer manufacturing system on their doorstep, basically.
With this bioreactor.
Well, with the bioreactor, because all you'd need is your solar panel, a simple thing to separate out the nitrogen from the air, and then you feed it into your bioreactor, and then you can extract out the ammonia.
Nitrogen.
So that's another area that's we're quite interested in exploring, is on-farm production. So the Haber-Bosch process is a really, really intense process, and so it needs to be carried out in very big chemical manufacturing plants. It's not suited to a distributed production. But a lot of the problems, well, a lot of the challenges is actually in the distribution and getting it out there. So the more you can have on-site production, those challenges
you don't have to worry about all these supply chains and lorries taking it all around the world. And so what are the little victories or little happy things, how you keep yourself motivated throughout the day?
So you're constantly producing results. So I think, and the group I'm in now is very good at celebrating little victories. So we sort of, every little result we get quite excited about.
Yeah.
And having other people get excited about it helps.
So it's that good community support.
So sort of a community support. And I think you do, because the challenge with science is you're working at such an aspirational long-term goal. You've really got to see the little wins along the way and the small steps in the process. I always find giving talks and things like that, where you present your results, actually is a really good opportunity for you to actually sit back and say, wow, we've actually done quite a lot. And this time last year, we hadn't even thought of that, and now look at all the stuff we've opened up.
Oh, yeah.
So I think that's always, for me, is a good opportunity for reflecting on just that you are actually making progress, even though it might feel like you're nowhere closer to the goal, but you've actually come quite a lot forward.
Yeah, you need to look at what you've done rather than forwards at what's left to do. Yeah. So thanks, this has been really interesting. And to conclude, we ask everyone to finish this sentence for us. To me, a scientist is someone who...
I'd say they're curious about the world and how it works and how we can make it better.
Okay. So not just curious about how the world and how it works, but also that trying to help people. You think that's an
I'd say
important
most
part?
scientists, particularly all the scientists I know, have this sort of, they want to see their work go somewhere. And even really sort of foundational understanding how things work is actually really important to being able to translate it.
Yeah.
So I think there is a need for that real fundamental research. But I think even those people are still doing it with the hope that someone else will pick it up and build on it.
Because what you're doing is like right on the applications, but you're saying someone who's more fundamental, it's still a long-term thing and their knowledge is still helping you.
I think it's Newton who said, if I've seen further than others, it's because I'm standing on the shoulders of giants.
Yeah.
So even the very applied science that CSIRO does is always built on a very, very solid foundation of other people's work. And that's definitely true in the nitrogenase area. There's been people who've been working at this for 50 years, just trying to understand how it works. And we're just adding our own little bit at the end. And I definitely would never take away from those people who were just doing what seemed like pretty simple, how does this enzyme work type of stuff.
Yeah, that's really great. And hopefully maybe in the next few years we'll start to see some of this technology starting to appear.
I do hope so. Yeah.
So thanks very much, Trevor. That's been really good. I've learned a lot of things.
Thanks for your time too. It's been fun.
Yeah. So I'm Ravi, and this is Mug of Science for Pint of Science Australia 2021.